Thank you to the Pompe Alliance for giving us the opportunity to discuss our new clinical development program. I'm Dr. Susan Sparks. I'm a clinical geneticist, and I'm a consultant with AeroBio in their clinical development. With me, I have Melissa Brooks, who is VP of Clinical Operations, who is in Philly with weather issues, so may or may not be able to connect. I'm also joined by Dr. Aslam Golkar Alpan and Lauren Knoll from LDRTC, who will also chime in when they come on. So I'm going to start, hopefully. Okay. Can everybody see this? Hold on.
Yes, it looks perfect.
Awesome. That's always the first challenge. I'd like to introduce a new molecule. This is ABX 1100. And I know that a lot of the discussions with this group has been on gene therapy or gene replacement therapy. This is a different type of genetic therapy. It's actually an siRNA. So this molecule, ABX 1100, has a protein, a centaurin protein that recognizes CD71 or the transferrin receptor, which is on muscle, and that is linked to a siRNA to GYS1 or glycogen synthase type 1. And the mechanism is that this molecule is given through an IV infusion. It goes to the muscle, and then it inhibits the production of glycogen through inhibition of the GYS1 mRNA. As we know, in Pompe disease is a deficiency of GAA, which cannot break down glycogen, and enzyme replacement therapy is the standard of care to work on the glycogen that is present in the lysosome. This molecule actually prevents the synthesis of the gray bar is enzyme replacement therapy. The blue bar is the ABX therapy that's given, and then the orange is ABX plus enzyme replacement therapy. And what you can see in the gastroc, the quads, as well as the diaphragm, there is decrease of glycogen per the milligram of tissue. And there's actually both together, the enzyme replacement therapy and ABX show a greater reduction between 70 and 80% of the glycogen in the Pompe mouse. So a study was started by Aero in normal healthy volunteers to show that ABX 1100 was safe. It was a single ascending dose given at 6, 12, and 18 milligrams. And then there were biopsies done before giving the dose and after the dose. Another group was given a dose, but with biopsies that were at week 6 and week 10. The reason these biopsies were done later is because there is time between inhibiting the mRNA, which then decreases the GYS1 protein, which then inhibits the glycogen synthesis. So it takes some time for the molecule to work. 2 doses were then given in part B, which is the orange graph, and the biopsies were done again at 6 and 10 weeks. And then currently there's the 2-dose study that has started in patients with LOPD. So I'll show you the data from the healthy volunteers. These are the same groups that were shown on the previous with single doses at 6, 12, and 18, and then two doses given at 18 milligrams a month apart. And what you can see is that there is a decrease in GYS mRNA as well as the protein, and you see that it's later. You can start to see some at 4 weeks, but more is seen at 6 and even more at 10 weeks. You also get more reduction when you have the two doses. So there's this long duration of action. So it's thought that after the two doses, there could be some maintenance dose that would be done quarterly. So in the normal healthy volunteers, there were no SAEs reported. Everything was tolerated well, and everything was looked at, because anytime you do a new study in patients, you need to look at their vital signs and the physical exam, the heart, as well as all the laboratory tests. They were all normal. Most of the treatment emergent adverse events were transient and mild, and mostly related to transient CK elevations. And this was most likely attributed to the muscle biopsy procedure. So I'm going to turn it over to Dr. Gokar Alpan to discuss further the safety and tolerability study of ABX 1100 in patients with Pompe.
Thank you, Dr. Sparks, and for this great introduction. And actually, this is, in a nutshell, is a substrate reduction therapy that is mediated by the siRNA. And it is one of actually the first kind, I believe, in the lysosomal disorders, and we're really excited to start this study. So I will just quickly go over the inclusion-exclusion and some basic, information about the study and about the site-specific questions. Please, actually, we have Lauren Knoll, who is the senior research associate, who is the supervisor for our clinical research group. So basically, in this study, late onset Pompe disease patients who will fit the criteria of inclusion exclusion that I'll review in the next slide will receive one dose of ABX 1100 as an IV infusion at day 1, and which will be repeated at day 29. So basically, they are 28 days apart. 2 infusions are administered. So obviously, the primary outcome measure is the safety and tolerability of ABX 1100, that is first in patient, but not in first human. So secondary outcome measures is obviously the pharmacokinetics in plasma and urine, whether and there is immunogenicity of ABX 1100 as measured by antibody formation in the serum, and also the pharmacodynamics by measuring glycogen synthase, 1 messenger RNA expression in the skeletal muscle. This is obviously done through this muscle biopsy. And if you have further questions, please refer to the clinicaltrials.gov site. The clinicaltrials.gov ID is listed on the slide. So if you review the key inclusion-exclusion criteria, so male or female LOPD patients that are adult, less or more than equal to 18 years of age, there is a body mass index limitation between 18 and 35 kilograms per meter square. Unfortunately, the patients who will not really follow these the body surface area requirements will be excluded from the study. Obviously, who is willing to go under the needle muscle biopsies and also have documented GAA deficiency and also with the molecular diagnosis. The patient should be receiving a stable dose of ERT that is FDA approved for a minimum of 6 months, and also capable of walking 50 meters with or without assistive devices. So if a patient can walk 50 meters with a walker, the patient will be admitted to the study. And also, the last but not the least, the forced vital capacity is more than equal to 30% within the past year prior to the study entry, not requiring invasive mechanical ventilation. Non-invasive temporary respiratory support, especially at night, is accepted. So the exclusion criteria includes basically if there is any other history of muscle biopsy within 1 month on day one. So if the patients are interested to be a part of other interventional studies that requires muscle biopsy, unfortunately, they won't be eligible for this study. Obviously, there is a history of concurrent illness such as coronary artery disease or cardiovascular disease, history of other GI, renal, or hepatic disease, and other condition that can interfere with the absorption, distribution, metabolism, or excretion of the drugs, and any other significant physical or other abnormality in the discretion of the PI, the patient may or may not qualify. So the other one is intolerance and difficulty of blood sampling through venipuncture, obviously positive pregnancy test for female participants, and if they are participating in another investigational trial, and also requires a wheelchair constantly and or invasive ventilation, they're going to be disqualified. These are the general inclusion exclusion criteria. And now actually I am gonna take a minute of our bio to introduce the LDRTCs. The observational study that we are actually trying to develop fluid biomarkers for Pompe disease. So this is a study 18 months that we are going to be doing MRIs with the Dixon technique to assess the fat water fraction, and going to be actually trying different biomarkers to do muscle staging or the involvement of the muscle at stages. And then we're going to be looking at the obviously the pulmonary function test and other clinical criteria. Also, we are planning to do a clinical staging of the disorder, which has, according to those results, which has not done before. So with this potential, it's an observational trial. We're going to be covering the travel expenses for the most, and also accommodation if needed, and also the MRIs will be performed twice as an additional service to the patient. So that's it. If you have interest, then we can actually screen the patient for both studies. Lauren Knoll is included here. Please contact her or me if there is any interest. Thank you.
Thank you, Özlem. Does anyone have any questions regarding the study or for Dr. Golkar-Alpan?
Can we were going to hold the Q&A to the end, if that's okay?
That works.
Yeah.
All right. Thanks, Heather.
Sure. All right. So can we, our next speaker is from Astellas, and he's going to talk about kind of the same thing with their clinical trial and some of the exclusion types of things. So thank you. And if you could just introduce yourself for us, I would greatly appreciate it.
Sure. Hello, everyone. This is Mark Walzer. I'm a global clinical development lead for Astellas Pharmaceuticals, and I'm joined today with Christine Brown, who is our patient advocacy support for the LOPD program, and Al Chang is also on the call as well, who's the asset lead and responsible for the global program strategy to help develop and deliver this exciting new program to registration. So what we're going to talk about today, we took a little bit different tact, and our program is a little bit more advanced. So we just wanted to highlight some important things about AT845 and give you an update on where we stand. And just first off, I do want to thank the community for giving us this opportunity to speak with you today. And we really appreciate your interest in hearing more about clinical trials, specifically gene therapy programs for Pompe. And know that your support is very much appreciated by Astellas. For a quick disclaimer, the safety and efficacy of the program I'm talking about, AT845, has not been established yet. There's no guarantee that it will receive regulatory approval or eventually become commercially available. It's currently not authorized for sale in any jurisdiction, and the discussion should not be interpreted as any kind of intent to promote the program or the product. So really, where are we with our trial? Our first clinical status for this. What we've done so far is we've shared some publicly available information at multiple recent conferences. And as you know, for those who have seen the data, we're currently conducting an investigational phase 1/2 gene therapy trial called Fortis. And the trial is using AT845 as an AAV-based gene therapy program, which would express GAA directly into the muscles in adult patients living with LOPD. The purpose of our phase 1/2 study is primarily focused on safety. And the secondary focus of the study would be to get some exploratory evidence of efficacy for this therapy. As of now, we've completed dosing for this trial. We've dosed a total of 11 LOPD adult patients in Fortis. 2 have been dosed at a lower dose of 3 × 10^13 VG per kg, and 9 at a higher dose, the highest dose in the study, at 6 × 10^13 VG per kg. So the study is currently not recruiting additional patients. We're looking forward to reading this study out in the March of 2026 timeframe. So we would have a lot more results as that comes out. A couple other things I want to highlight about the program to date. Many of you might have seen that it has received the FDA-granted RMAT status, which stands for Regenerative Medicine and Advanced Therapy. This program allows us to work closely with the FDA to help speed up development, speed up the review process, have more meetings with the agency, and it is focused on programs that are intended to treat serious or life-threatening conditions and have some signs of early clinical evidence showing potential to address some of these unmet needs. So what does this mean for Astellas? As I mentioned, Astellas has now the ability to have a little bit more frequent and timely communications with the FDA, and help us develop plans and studies to eventually show a safe and effective potential in LOPD patients and help accelerate the review process of our program as well. In terms of the Fortis data, what we've presented recently at the World Symposium this last year, we presented a data cut based on September 30th, 2024 data. And there we presented that 9 total patients had received gene therapy at that time. Since then, we've dosed 2 more. Of those 9 that received it, we shared information that 6 of the 9 had been in the study for at least 36 weeks or more. So we have some longitudinal data in these patients of at least up to 3 years here. And overall, the gene therapy infusion was well tolerated, with several possible side effects, though, were observed. Those include infusion-related reactions like nausea. Other common events that we saw were increased liver enzymes, which can lead or can indicate to liver damage in these patients. Two participants, did develop medically significant elevations in liver enzymes. However, these were treated with IV steroid infusions, and all the cases improved with steroid treatment. We'll plan further results in Fortis to be released in early 2026 at various conferences. And we look forward to that. In terms of the efficacy, we did want to highlight that the presentation at Worlds did show that the 6 patients dosed with the higher dose, the 6 × 10^13 dose, 5 of them have stopped ERT treatment. They were previously on biweekly ERT treatment. And they had stopped ERT after receiving the gene therapy without showing any clear evidence of disease progression over the time frame that we have been monitoring these patients. As of today, the longest time off ERT at this high dose group is approximately 3.5 years, and the lower dose group we've been monitoring these patients for about 4 years. So that will stop, and again, we're not recruiting into this trial, so I don't think we'll go into in-depth details about the inclusion-exclusion criteria at this time, but we would refer you to the clinical trials website link that we can put in the chat if you're more interested in hearing more about the trial design.
Thank you. Um, it's so we can and if there's nothing, if we have nothing else from our presenters, Arrow or Stellis, we can move into the Q&A portion. Again, if you don't feel comfortable speaking, you can put your questions in the chat. Carmen and I will be monitoring that.
Maybe while we're waiting for people, we had some questions come in from the earlier meetings that we had. So this is the third in our series, and the first one was sort of an overview of approaches to therapy in Pompe, with a focus on sort of genetic medicines, but broadly approaches to therapy. And then the second one was really hearing from people who had participated in clinical trials about their motivations, their experiences, sort of their advice. And so throughout those, we have collected some questions that came in that were more appropriate to hold and ask you, the therapy developers, instead of trying to answer ourselves. So I see that
And we actually just, sorry, Carmen, we did have a question just come in. It says for both trials, is there a plan to expand to LOPD?
I mean, I can speak for our trial that we're in this trial is in LPD, the Fortis trial, a phase 1/2 early trial, again, on safety and efficacy. I can say in terms of the next steps for us in the LOPD population, we would be looking to start a phase 2, potentially start a phase 2/3 trial where we would expand into a more broad LOPD population and much more numbers of LOPD patients. That would be based on proof of concept results coming out of the Fortis trial in March of 2026. We're currently assessing sites for the phase 2/3 study. Of course, we have some phase 1 sites that we used in Fortis, and we're assessing whether we can include those in the phase 2/3 as well. And then we're going much more global for the phase 3 study. We're looking at sites in Europe and further sites in the US, as well as Taiwan and some of the other countries. We can come back and discuss more of the trial logistics when we get closer to designing the phase 3 trial. Right now, we're just in the process of trying to understand what the inclusion-exclusion criteria would look like and how best to design the trial with input from the patients, of course, and then regulatory bodies as well.
So maybe we should
go ahead.
I was going to say, maybe we should pause and make sure that the Arrow team also discusses that sort of LOPD versus IOPD and next steps.
Sure. So as we know, ABX 1100 is an investigational product. It is the first time that we're going into patients, and because of that, it needs to be adult patients. And it starts in adult. All clinical trial studies start in adult patients. And at this time, there isn't plan to expand, as we're still just trying to understand the mechanism of action. I will answer the other question. So as far as international, this study is open in Canada as well as the US. And the other question about symptomatic versus non-symptomatic, patients do need to be on a stable dose of enzyme replacement therapy. Otherwise, there is no criteria as far as they can't be too severe. So it's the severe end that is excluded rather than a milder end.
Hi, doctor. Hi, how are you?
Hi.
So I didn't want to type it out. I figured I'll just talk about it. So with the ABX 1100 for adult, and then obviously late onset Pompe, would it be a study where they're going to be taken off ERT, or it's incorporated with the ERT?
So at this time, this first study, which is a phase 1 study, patients remain on enzyme replacement therapy throughout the study, and then they're given just the 2 doses. This is what we call a proof of concept study to see if it works. So there's no plan to take anybody off of enzyme.
Okay, thank you.
I may have one comment on Susan's whether, or actually the question is whether symptomatic and non-symptomatic. We'll be happy to see those naive patients under the observational study. Actually, in my experience, the patients, even who are not symptomatic, they are symptomatic with the muscle involvement. And if they qualify for enzyme therapy, we can ascertain that we are the first site to open it. And I am sure that we can actually get some of the patients, if they qualify, within 6 months to the study, if they're interested.
Mark, can you answer that same question? And then we'll go, there was another question and we'll go back to that one.
Sure, the question on patients coming off of ERT. Yeah. So in the Fortis trial, patients were allowed to come off ERT following 16 weeks after the gene therapy. Gene therapy will take some time to develop a GAA and express in the muscle. And so we allow patients after 16 weeks to come off of their ERT, but they were on ERT. They were ERT experienced patients coming into the trial at least 2 years of ERT, a stable dose for at least 6 months. And then after 16 weeks after gene therapy, they were allowed to come off of ERT. And then we monitored, of course, closely safety endpoints for those initial couple months as they came off, and then continued, of course, monitoring for the months following. For the naive population, we don't have any data yet in naive patients, so patients who have not received ERT. We are assessing if that can be a population that we can go and look at that population in additional future trials as well.
And then we had another question. What metrics are you using to determine that gene therapy tested patients do not need to be on ERT? And are you looking at muscle GAA levels?
Sure. Thank you for the question. Yes, it's a very important part of our Fortis trial is being able to monitor the safety of these patients as they come off ERT if they choose to do so. And those assessments are primarily being done using forced vital capacity, 6-minute walk tests, and also, for those familiar, the RPAC tests and some of the other functional assessments more specific to Pompe disease itself. Patients come in weekly visits after they come off of ERT for, I think it's the first couple months as they come off. And those assessments are done very routinely. So we make sure that these patients are not decompensating their disease rapidly. And if that was to happen, we would, of course, have an opportunity for these patients to go back onto ERT in the Fortis trial. We are looking at GAA activity and expression of the GAA protein in the muscle using biopsies in these patients. We take 2 post-dose biopsy time points. We take a sample, the latest being 1 year after gene therapy dosing, and compare that to the sample taken prior to gene therapy dosing.
Do we have any other questions before Carmen goes back to the questions we had from the previous meeting? We have one just come up. It says, this is for Arrow. My understanding is that glycogen is used for energy by muscle and that Pompe stops excess glycogen being removed. If the muscle is stopped from producing glycogen, do they still get sufficient energy to work?
So this question for the GYS1 knockdown or inhibition of the mRNA, it's not absolute. It's up to 80% of a reduction. So there is still glycogen for energy.
And the next question is, do you envision that there would be redosing at some point after initial dosing?
And is that for the Arrow?
It doesn't say. That was just a question that came in.
So as far as Arrow, the study that we have ongoing right now is for 2 doses, what we consider a loading dose. As we evaluate the study for safety as well as the knockdown of the mRNA, we will do future trials to look at future dosing regimens.
I can speak to the gene therapy space as well. I mean, that's a big question in the gene therapy, AAV-based gene therapy space. Just to provide a little education, when patients receive an AAV-based gene therapy, the body usually develops antibodies to AAV if they haven't had that exposure before, and antibodies to the virus. And so having very high levels of antibodies in your body over months and years, even after getting gene therapy, there's a potential that an additional dose of gene therapy would not be as efficacious because the antibodies would attack basically the new gene therapy that was given, the new AAV that was given. So the field in general is trying to come up with opportunities to dampen the immune system for the second dosing, or tackle it different ways to allow patients to have the ability to have more than one dose of AAV-based gene therapy. But currently, that's not something that AT845 has assessed yet. And the field in general, I think, it is trying to understand the science behind that right now, and whether or not it's safe and be effective as a second dose.
Thank you. How long is the RNA therapy in the muscle? If it doesn't agree with our bodies, what is the half-life in muscle? This is for Arrow.
Yeah. So we're assessing the half-life in muscle. The half-life in the body or in the blood is very quick. It gets taken up by the muscle. But we have seen in the preclinical models and things with the non-human primate as well as the mice that it does return to normal. So I can't say the exact half-life in muscle, but it does return to normal.
Thank you. Were you going to say something, Carmen?
Nope. I'm just
Okay.
noticing questions coming in and monitoring them.
Will LOPD patients be included for any of the new therapies after clinical trials if they cannot get ERT treatment? The idea that not being eligible for ERT excluding you also from gene therapy or substrate therapy is quite sad. I understand for clinical trials, but I'm referring to after that if these are successful.
Mark, I think you just put yourself on mute and started talking.
Yeah.
Sorry, my apologies. I'm happy to start. We absolutely see the unmet need in patients who haven't even started on ERT yet. We understand that ERT is even being started earlier and earlier, but there are patients who are naive to that, and we would not eventually want to have them excluded from the gene therapy study. However, we need to study those patients first. We need to understand the safety and efficacy in that population as well. But in general, we would like to target as wide a population of LOPD patients as would be safe and efficacious for the program.
For the Arrow product, at this time, because it is a different mechanism of action than the enzyme replacement therapy, the thought is that this therapy could be something that is used to decrease the burden, but enzyme replacement therapy would likely be needed to clear the existing glycogen. That said, future studies could look at naive patients, just as Mark just mentioned. Those are future studies after these initial ones are completed.
For the gene therapy, do you anticipate that patients would need to be on immune modulators due to the antibody production?
Yeah, I'll reiterate for Fortis, patients were on immune modulators, so they were started at day -1 prior to dosing of the AT845, and they continued on prednisone or prednisolone for up to 16 weeks, and some of that was the last 8 weeks was a taper. So patients eventually then came off. Different patients were on the immune modulatory therapy for different timeframes based on what kind of adverse events came up in those patients. And so, for example, some of the liver enzymes, as I mentioned, were elevated in patients, and the immunomodulatory therapy could help decrease those liver enzymes in those patients. So they continued on immunosuppression longer than was traditionally set in the protocol for some of the other patients. Long term, they would not be, we would hope, the goal is that they would not be on immunomodulatory therapy for long term after they receive gene therapy. But at least what we've seen in Ford is some patients up to 6 months.
What is the name of the virus that is evaluated for antibodies? I was eliminated from a trial because I had antibodies. Wondering if you use a different virus or the same.
So for our exclusion criteria, there is a cutoff of the level of antibodies to AAV, adeno-associated virus. And ours specifically is AAV8. There are different serotypes of the virus. Different gene therapies could have different serotypes, 1 through whatever we're looking at 8. So we would be specifically measuring antibodies to AAV8 in our trial, and some patients, would have some low baseline levels just based on exposure in the environment to natural AAV8 that's occurring in the environment. If they have very high levels to AAV8, they would be excluded at this time from, or were excluded in the Fortis trial, and we're evaluating what levels of antibodies would be allowable in future trials for AT845.
This Stuart says he appreciates that this is a difficult question, but if all trials continue well, what kind of timeframes should we imagine until approval and widespread availability for both treatments?
I can ask for or answer for Arrow. This is the first study in humans, and as we know, clinical trials can take several years to get to an approval. I can't predict at this time, since we're so early in the development, how many years it's going to take to get where it would be widely available. But of course, that is the ultimate goal.
Yeah, I think I would echo Susan's comments. It's difficult to set a timeline now. We, again, having RMAT give us some abilities to accelerate as much as possible and working with some regulatory bodies. But just hopefully starting upcoming phase 3 trials, those take a few years, and then there's time needed for regulatory approval and review as well. But unfortunately, I can't give you a date right now.
Would participation in either of these trials exclude a patient from participating in further gene therapy trials?
For us, for the Fortis trial that was ongoing, it did not allow for patients to have been exposed to other gene therapy trials. We haven't identified what the inclusion-exclusion criteria would be for the phase 3 trial coming up. I will highlight that most AAV-based gene therapy programs have exclusionary language around, again, antibodies to AAV8. And if you might have been exposed to AAV8, previous gene therapy trials of AAV8, it's possible that your antibodies could exclude you to future trials. But it would really be a patient-by-patient basis. And each program, each protocol is different. So I wouldn't want to give a generalization that if you've gotten gene therapy once, then you could never be treated again. I wouldn't put that generalization.
For the Arrow product, it is an siRNA. And because of that, if you're off of it, there would be, 5 half-lives, and then you could participate in another therapy. But because it's a different type of therapy, it should not exclude you from a gene therapy trial in the future.
Can I chime in real quick?
Sure.
I'm just going to say across the board for studies, and this is not just Pompe specific, but across the board, if you have had a previous gene therapy treatment, that in most cases will exclude you from any further treatment study within that disease group. That is the common inclusion exclusion right now across the board for all studies. That may change over time as years go by and we learn more. But that right now is a pretty standard exclusionary factor for any study that is being proposed, that if you've had previous gene therapy, you are excluded from participating in the treatment study.
Thank you. We've got about 10 minutes if anybody else has questions. I actually do have a question. You talked about, Mark, you mentioned the elevated liver enzymes. I myself have elevated liver enzymes, and I know other Pompe patients do as well. Does that exclude, would that exclude a person from this particular treatment or participating in future clinical trials, if that's a side effect?
So I'll broaden it that generally, of course, gene therapy programs are concerned with liver toxicity. That's been seen in multiple other AAV-based programs. So there will probably be language in most gene therapy studies around patients with elevated transaminase, ALT, AST levels. There will be language in there around patients who may be excluded for having certain levels of liver disease. So it's absolutely something that is traditionally in exclusionary language. The question is really like how elevated are your levels? So at each individual patient, there's obviously a gradation of what would be allowed in a trial. If they have levels that are acceptable to the trial, they could be in, but if they're too high, then they would likely be excluded from those trials.
Thank you. We have a few more minutes.
I think all of the questions from the first couple of sessions were asked, with the exception maybe of one that I suspect probably only is really applicable to the Astellas team. And it's really about like the expectations, your anticipation expectations for the next clinical trial. And you touched on this a little bit, but I think you said that the readout from this study is expected in March of 2026. So how long would you expect it to be from that readout to starting enrollment for the phase 2/3 study? And is that study expected only to be in the US or to be outside of the US as well?
So the second part of your question is the easier part, right? So that will be a global study that we're planning right now. So we would have US sites, and as currently planned, we would have multiple sites across Europe and again, Asia and so forth. So it'd be a global study at that point. When that study starts is difficult to say right now. We have to go through multiple steps of regulatory approval, so forth. But I think we're looking forward to the March 26th readout, and then putting together some timelines of when we would be able to start that phase 3 study. Sorry, I can't be more specific on the study.
That's right. I suspect Susan can even be less specific based on what she said before about this being so early stage. If you want to tell me I'm wrong, I'd be happy to take it.
No, I was just going to say I can repeat what I
right.
No, it's so early. I don't know. Yeah, so that's where we are.
I think we're definitely committed to, coming back and speaking with, multiple Pompe communities, patient communities, and being, very engaging and very open with what these studies will look like and what kind of inclusion exclusion criteria could allow into the study. So we want to definitely have this dialogue and make sure you're aware of the most up-to-date information so you can make decisions for yourself and your own treatment.
Thanks, Mark. I completely agree with that as well. We want to be partners with the community, and we will be as transparent as we can. And as more data comes along, we will be happy to share it.
Perfect. Heather, we only have a few minutes left. Do you want to just pop up that slide? So I know one of the things that we've talked about with the group before is that our team, the RareX team at Global Genes, has been working with the Pompe community to build out this Progress for Pompe study and to.
I didn't get that slide.
Oh, you didn't
Carmen.
get that slide from Caitlin?
Mm-mm.
Caitlin's still
I
on.
don't think so.
Maybe if she has the ability to share, she can probably share it. We can also just put links in the chat. I'm sure that Caitlin or Tina could put a link in the chat. So for anyone who hasn't signed up yet, it is a great way to find out about upcoming clinical research opportunities, clinical trials. We are really committed to continuing to work with the therapy developers and try to stay abreast of changes. And by joining, you can tell us, this is something I'm really interested in, and I'd like to be notified when there's an opportunity. So it's a really great way to stay in touch that way. And then I think Tina will probably put in the chat, if she hasn't already, that she's actually hosting kind of a little sign-up party tomorrow morning. So Saturday morning, when you're not working and you're in your jammies with coffee, you can come and meet some other people and sign up together. Oh, there's another question or two.
Yeah, one more question. So first, the meeting is being recorded and it will be available for view once we have it edited. And then this is for Arrow. Are you looking for subjects still for trials on ABX 1100?
Yes, the study is open. As Dr. Gokar Alpan and Lauren have mentioned, they are a site. So you're welcome to reach out to them if you have questions about enrollment.
Yeah, we can take any patient from anywhere. We can actually have it traveled, the patient, and it's no strings attached. But I know some sites require certain insurance qualifications that our clinical and research operations are completely different. So basically, we don't look for anything except for the study qualifications. So let us know if anybody's interested, and if they don't have the finances to travel for the first time, we can look into that too from NORD or other patient organizations to help with the travel.
Thank you. Caitlin just put a link in there for the the registration for the project. Yes, thank you. So with that, we'll close out for today. It's just about 2:00. I would like to thank our speakers from Arrow and Astellas, as well as Carmen with RareX, and Anne, who's with the Pompe Warrior Foundation, who's not present today. But Pompe Alliance does not do this by ourselves. We do this with Carmen and RareX and Pompe Warrior Foundation, and so we really appreciate their collaboration as well. And we're happy to have the Arrow and Astellas come back at any time that they want to share more information with the community. We're definitely happy to do this again. Excuse me.